So central dogma, DNA to RNA to protein to trait. And again, we have sub-steps. DNA to RNA, we call transcription. RNA to protein, we call that translation. Expression is protein to trait. Yeah? Okay. Questions? Okay, so transcription, let's look at it. Transcription, at the level we're looking at it now, is pretty simpler. Again, we know that messenger RNA carries information from the DNA to the ribosome. That we knew already. 3 DNA or RNA bases we call a codon. 3 nucleotides is 1 codon, codes for 1 amino acid. Again, that's review. We talked about the universality of the DNA code, yada, yada, yada. So here is a picture that I want you to understand. This is the process of transcription, yeah? And we can see here DNA being pulled apart, just like it is in replication, but instead here, it's not being pulled apart all the way. It's just being pulled apart a little bit, yeah? And as it's pulled apart, messenger RNA is made off of one of the 2 strands. Does that make sense? And so A matches with T, G matches with C, A matches with U. Yeah, does that make sense? G with C, G with C, T with A with U. Got it? Because uracil replaces thymine in RNA. Okay, got it? Right? And so what does this? RNA polymerase, right? DNA polymerase does replication. RNA polymerase does transcription. Yeah? And all it does is read one side of the DNA and match it up with RNA bases to make a single strand of RNA. Got it? And as it's done, the DNA closes right back up, right, as it goes through, right? Here it's going left to right, and as it goes, the DNA closes right behind it. All right, so again, the shape of this thing and how this thing works is pretty important. It got a Nobel Prize. Right? So here are the pictures from your textbook, yeah? We pull apart the DNA. Here our RNA polymerase is an oval instead of a sort of a torpedo shape. And our RNA isn't in pink, it's in sort of turquoise. But basically, it's all the same thing. We read along. The messenger RNA gets longer and longer and longer. And then once we've found the end of the gene, we let go, yeah? So transcription binds, DNA binds to the promoter, yeah, and ends at the terminator, yeah? So, so again, RNA polymerase lands in a particular stretch of the DNA before transcription starts. Can you see transcription starting here, where the pink line is just beginning? But it lands in this green spot. This green spot is called the promoter, yeah? Right? And the promoter is a piece of messenger or a piece of DNA that RNA polymerase really likes to stick to. Yeah? Does that make sense? So it lands on the promoter, starts reading the gene, and then it goes to this orange sequence, and the orange sequence is called the terminator. Yeah? And the terminator is where the RNA polymerase lets go after the gene is done. A terminator is not the same as a stop codon. Yeah? And I'm sorry, I have to pronounce it that way because I'm from California, you know, so we have to. The Gavinator was the terminator. So, sorry. So if we go all the way back through all this stuff, we can see here that a stop codon is not the same as the terminator, right? The terminator is for DNA. This is for RNA at the ribosome, yeah? So it's slightly different, okay? All right. And it's just a piece of DNA that RNA polymerase doesn't like very much, okay? So we bind to the promoter, we stop at the terminator, and that is transcription. Guys good? Okay. Translation. Deep breath. So transcription, fairly simple. Translation, a little more complicated, yeah? Okay. So, translation, messenger RNA to protein, yeah? So let's look at how that's done. Messenger RNA information is translated into polypeptides at the ribosome. And again, polypeptides effectively means proteins, yeah? And proteins are made out of amino acids, yeah? A bunch of amino acids all in a linear row makes the primary structure of a protein. Guys with me? Questions? If you got questions, I'm good to answer them, yeah? Okay. So, what color are tRNAs in this picture? Purple is correct. What color are amino acids in this picture? Green. What color is messenger RNA in this picture? Orangish red, sure, depending on how you see that color, yeah? And what color is the ribosome in this picture? Blue. So in the same way that DNA polymerase's job is to match DNA pieces to a DNA template, yeah? In the same way that RNA polymerase's job is to match RNA nucleotides onto a DNA template to make a messenger RNA, likewise, the ribosome's job is to match up messenger RNA codons to what are called tRNA anticodons. Okay? You guys see that? And there's 3 bases here that are matching with the codon, each codon of messenger RNA. Yeah? And all of those, the DNA polymerase, the RNA polymerase, and this guy are all kind of acting like the head of a zipper, right? And reading along, yeah? Does that make sense? So, the ribosome reads the information and matches up tRNAs to messenger RNA codons, yeah? Okay. And each RNA is holding a different amino acid, right? This one is holding glycine. This one is holding phenylalanine. These ones aren't holding anything yet. They're going to have the amino acid put on there first, yeah? Does that make sense? Okay. So, again, here's another Nobel Prize for chemistry, structure of the ribosome, and these are interesting interviews with Ada Yona. How many women Nobel Prize winners have there been? Yeah, so the answer is, I think, significantly fewer than there have been deserving women scientists. And that's been sort of a sensitive issue. So there was an interview. These are interviews about how basically, yeah, women can be Nobel Prize winners too, apparently, yeah? To some degree, that's up to the Nobel committees, right? Which are usually not women, and that may have some influence on their judgment. All right, anyway, so eventually people straighten things out, and so 2009, this person got a Nobel Prize along with these guys. There's the structure, and again, knowing the structure of this thing is pretty important, yeah? So tRNA is the interface between messenger RNA and protein, and so there's a couple of parts of tRNAs that you should know in order to understand how they work, yeah? The 2 parts, one we've mentioned briefly, the anticodon, yeah? And the anticodon is down here, right? Anticodon, can you infer some information about what that does? It's the reverse of a codon, yeah? Does that make sense? So, these are made out of RNA. So if a codon is AUG, then what should the anticodon be? GAC. Does that make sense? Right? And then, what should this guy be holding? It should be holding a methionine RNA. Or excuse me, should be a methionine amino acid. And I should draw that amino acid in another color, yeah? I'll draw that in green. that clear? Because if we look at AUG on our codon chart, right, the amino acid it codes for is methionine, and it also says start, right? So nearly every protein in prokaryotes and eukaryotes, at least when it's being built, starts with methionine, yeah? Because that's the starting amino acid for almost every protein. Sometimes that amino acid gets clipped off, I think more in eukaryotes than in prokaryotes, but that's neither here nor there. Okay, so we've learned about the anticodon. There's one other part to a transfer RNA that we care about, and that's called the amino acid attachment site. Amino acid attachment site. And what do you guys can infer about what the amino acid attachment site does based on its name? That's where amino acids attach, yes. Does that make sense? The job of a transfer RNA is to bring amino acids to the ribosome, yeah? And the job of the ribosome is to assemble those amino acids according to the information on the messenger RNA. Guys got it? Right? And so in order for them to make sure that they're attaching the right amino acid in the protein in the order, right? Remember, the order of amino acids really matters. That's the primary structure that determines the whole shape of the protein. Yeah? You guys remember that? All right? So, in order for that to happen, the right anticodon has to match with the right messenger RNA codon. Yeah? And it has to be carrying the right amino acid. Right? And so if I drew it here, the anticodon would be AUG, would be GAC to match with AUG, the start codon, and the amino acid would be methionine. I wrote meth, and I was like, oh, let's not bring that into here. Okay. All right, is that clear? You guys understand? All right. Good, good. So, how do we make sure that we put the right amino acid on the right tRNA? Well, each tRNA has an enzyme that adds the amino acid on there. Yeah? So, can you see the tRNA? What color is the tRNA? Let's look at the shape of it, yeah? There's what a tRNA looks like. What color is it in this picture? Sort of red and yellow, yeah? This red and yellow structure here, you see the similarity between this structure and this green thing here? Yeah? All right. What is the big blue glob? That's an enzyme. What catalytic reaction is it doing? It's catalyzing the reaction of attaching this amino acid, shown in green, to the amino acid attachment site. Does that make sense? How many active sites on this enzyme? I would say there's one active site right here. There's also an allosteric site. Can you see where that allosteric site is? David has got it. He's pointing at it. Where is the allosteric site? If you're pointing here, you're correct, yeah? And why would there be an allosteric site here? You got to make sure that this is the right anticodon to put on the right amino acid. Does that make sense? You guys following along? This is complicated, so if you have questions, if you want me to repeat things, could you just repeat that, and I'll try to say it in different words, yeah? Could I repeat that? Yes, I could. All right. So, in order for this process to work, yeah, we got to make sure that we put the right amino acids in the right place, yeah? All proteins depend on their structure, yeah? Guys with me? Right? And so, where do we get the information for how to make the protein? The DNA, right? But we're out here in the cell. We're not. The DNA is way the heck over there. What do we use as the surrogate for the DNA? The messenger RNA is the messenger carrying the DNA's message. Yeah, you agree? Right? So So as far as this ribosome is concerned, the information from making the protein is coming from that messenger, yeah? The messenger RNA. And if the messenger is saying that I've got, what's my code for alanine? Hold on. Here it is. Alanine. ALA. So GCG, yeah? So if this is GCG, then what's this got to match? G with C. C with G, and G with C. Right? And what's it holding? It's got to be holding an alanine in order, right? If it's holding the wrong amino acid, then everything gets all messed up, yeah? Well, let's go forward a little bit again. GGC. So the next one says GGC. GGC. Right? And that's the amino acid code for glycine, right? And so effectively, when this thing has GGC, it's saying, okay, ribosome, put a glycine there, yeah? So how do we make sure it puts a glycine there? Well, this thing has to have what? C, C, G, yeah? And it has to be holding a glycine, yeah? Well, how did this glycine get on this transfer RNA? Here's one that doesn't have an amino acid on it, yeah? And once they're done, the tRNAs have to leave and the protein has to stay. Do you see that? So this guy here on the left, this guy here, where's he going to go next? What's his job now? He's got to go pick up another amino acid, yeah? Right? And he has to pick up the right one, yeah? Well, so how do we make sure that we put the right amino acid onto the right tRNA? Well, we check your anticodon, yeah? Does that make sense? Right? Okay. How many people benefited from that second explanation that somebody asked for? All right. So what do you say to Kalsang? What do you say, Kalsang? There we go. Got to have manners around here. Okay. Is that good? Again, I do my best to explain things, but the fact that people don't always understand every word that comes out of my mouth is pretty I blab a lot, yeah? So if you want a second explanation, you can just ask me to repeat something and I'll do my best, okay? Okay. So, translation, right? Transcription, DNA to messenger RNA. Translation, RNA to protein at the ribosome, yeah? Okay. So, here's basically, this is a dynamic process. This is a process that involves motion. We can look at it with our little flip book here, and then maybe we can watch a video or something, okay? Does that sound good? Guys with me here? Okay. So, prokaryotic translation, we'll go through this fairly fast, and then maybe we'll see it again, and then so on, yeah? So, step 1, ribosomes assemble around the mRNA start codon. You guys see that? Guys with me? So, there's the messenger, there's the methionine tRNA, right? And the methionine has to be added by an enzyme, yeah? And then this guy, UAC, matches with the AUG of the start codon, yeah? Right? Okay. So, there it is. Our ribosome is assembled and ready to go, holding on to the first amino acid in our future protein, yeah? The next amino acid, the messenger RNA says we need a leucine in the sky. So UUA matches with the tRNA that carries leucine, yeah? And it comes down and matches up, yeah? Then this methionine covalently attaches to this leucine. Yeah? Is that clear? And then the next one comes on, and then the next guy comes on, and so on, and so on, and so on. And you can see our our polypeptide chain, in other words, the primary structure of our protein is getting longer and longer and longer, the further along the messenger RNA the ribosome reads, yeah? Ribosome reads the messenger RNA, grabs tRNAs to match up, yeah? Guys with me? And makes the amino acids one by one by one by one until that protein is completely made. When we get to a stop codon, what tRNA goes in there? No tRNA goes in there, and so that's where the stop codon signals completion of the polypeptide. Yeah? And then the ribosome disassembles, the new protein folds up into its secondary structure and its tertiary structure and sticks with the other polypeptides to make the quaternary structure, and then goes and does its protein job. Does that make sense? And this ribosome is going to have to assemble around another messenger RNA and make another protein sometime. Yeah? Does that make sense? Shall we see it again? See it a little faster? Let's go back. Right? It's like we're looking at a little flip book, yeah? So, ribosome attaches around a messenger RNA. The methionine tRNA shows up and binds to the first codon. Then the next tRNA shows up with leucine. And then the next, and then those two amino acids link together. That frees up this tRNA, right? This tRNA is ready to float away. Bye-bye tRNA that's empty. Where's this guy going to go? He's going to go to another enzyme to get another amino acid stuck onto him so he can go do his job again. Yeah? Is that clear? Right? All day long, this guy drops methionine amino acids into proteins and then goes and gets more methionine from another enzyme. Yeah? Guys got that? Right? Spent tRNAs dissociate. The polypeptide chain continues to grow in this manner. The whole thing leaps off and attaches to the next one, right? So AUG is the start codon, but sometimes we just want a methionine anyway, yeah? And then pretty soon, we're done with our protein, and our protein goes and does its job. It folds up into the right shape, and then the ribosome goes and starts again somewhere else, yeah? Guys got it? Do you guys want to see it in video form? It might be helpful, yeah? Okay, so we're going to stop